All right, so this is where we left off then. We were talking about this theme number 5, which is evolution, and that's biology's core theme. Now, evolution explains both the diversity and the unity of life on Earth. It's kind of crazy, isn't it? You've got one theory that can explain both, why life is so diverse, yet why we have commonalities, features common to all living organisms or organisms within a group. And Darwin originally explained this, put his explanation forth on his book On the Origin of Species by Natural Selection. So we're going to have a closer look at that. Now, it's difficult to sort of distill down Darwin's theory of evolution, but we can, and he made 2 major points. All right? That species showed evidence of what he termed descent with modification from common ancestors. So to maybe slightly reword that, Darwin said that species today show evidence of being descended from ancestors, but that over evolutionary time, the species of today are different to the ancestors that they are descended from. Okay? And that they've become better adapted to their environment. Those adaptations, those modifications, usually make them better adapted to the environment they're in. And then he proposed natural selection as the mechanism that caused the change, and that natural selection explains how we become better adapted to our environment. So is everybody okay that what descent with modification is? Yeah? Can anybody give me, I don't know, I guess an example of, look at a feature that you have and maybe take that feature and say, well, this kind of it is an example or explains, you know, how we're descended from common ancestors that supports that. What about teeth?
Like we have canines.
We do have canines. They're quite reduced.
Mm-hmm. So, like to eat meat and everything like that.
Okay.
Herbivores and everything.
Think about if we're descended from an ancestor, presumably there are other organisms on the planet that are descended from ancestors. In fact, we all are. Right? What would be evidence that us and, say, closely related, similar organisms to us, maybe were descended from a common ancestor? Evidence that we came from one ancestor versus we had, I don't know, many different lineages, that we were descended from many ancestors. So what about opposable thumbs? What about them, though?
No other creatures aside from primates seem to have them.
All right, so all primates have got opposable thumbs. Yep. Is that just coincidence? All primates also have a vertebral column. All primates also have hair. All primates share many characteristics, don't they? Is that coincidence? So Darwin said that it's not coincidence. We share those characteristics because all primates came from an ancestor. And that ancestor had all of those characteristics that us and other primates have. So if we're descended from a common ancestor, then chances are we're going to retain some of those characteristics. But as we change over time, we're also going to modify some characteristics that we got, maybe acquire new ones, and lose some characteristics. Does that make sense? So when you share characteristics, when you have characteristics in common with other organisms, that's really good evidence that you have a shared common ancestry. Okay? All right. Okay, so I want to take you through sort of Darwin's explanation and Darwin's reasoning as sort of how he came up with some of his ideas. And these were based on some of the observations he made. So as I'm sure you know, Darwin was around in the 1800s. One of the pivotal points in his life is that he went on a 5-year journey on a boat called the Beagle. And, sorry?
Was it Czechoslovakia?
He did go to the Galapagos. In fact, he went almost all around the world, lots of different places. So he went to all these different places and he looked at the wildlife and the plants and the animals and the fossils. And as he was looking at these, he said, oh, wow, he said, there's a lot of different species and they're very different. He said, but they also share some common features. And so on this voyage, he collected a lot of specimens. He started to think about how can we have such a variety of life on Earth that yet share common features, features that are common to groups or all organisms. And of course, at the time, the idea is that all organisms on the planet were created by God, right? So, and you couldn't question that. But Darwin did. Darwin said, wait a minute, some things just don't add up and don't make sense. There's more going on here. So I'll sort of take you through his observations that led him to make the inferences that he did. So firstly, Darwin said, well, individuals of a species vary. And that's not a big leap, is it? You are individuals of a species, you're all Homo sapiens, and look at the variation just within this class. There's tons of variation, isn't there? Think about every saguaro cactus out there. There's variation among saguaro cacti. Think about dogs, the variation among dogs. So within every species, there's variation. Sometimes that variation is huge. Sometimes it's quite small and quite subtle. But there's variation of individuals within a species. Is everyone okay with that? That's not rocket science, is it, really? It seems quite intuitive. All right, but let's just think about that. Variation within a species. Darwin then said, and then noticed, that a population is able to produce far more offspring than actually survive. Okay? So the reproductive potential of any population is much greater than what actually survives. Is everyone okay with that? Doesn't matter what species you talk about, they're able to reproduce and produce more offspring than what can survive, at least in the long term. Is everyone okay with that one? Again, seems quite straightforward, doesn't it? All right, now let's think about this next one. Here's what Darwin said, is that organisms are typically well suited to their environment. He said they're very well suited to their environment. Again, it's just another observation that he made. So now let's sort of think about those observations and let's sort of mull them over and see what we can deduce or conclude for them. So here's what Darwin concluded. Darwin said the best adapted, so the ones that have the best variations for that environment, they're more likely to survive and reproduce than the ones that are less well adapted. So the well adapted variations are more likely to survive and reproduce than the less well adapted variations. And if you're more likely to survive and reproduce, you're more likely to survive, you're more likely to pass on those advantageous characteristics to the next generation. Does that make sense? Seems quite intuitive, doesn't it? Yeah. And so individuals with these advantageous traits or the advantageous variations, they generally increase in number within a population. So over many generations, we get the accumulation of advantageous traits or characteristics. And therefore, over many, 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 many generations, individuals in that species end up changing. They change over time. All right. Everybody okay with that sort of the process, the thinking behind that? Yep. All right. So natural selection then, which I've just described, that is really natural selection. That was Darwin's mechanism of evolutionary adaptation. Okay. Darwin said, well, organisms become better adapted to their environment through the process of natural selection. So here's an example that's in your book. It's kind of a good example. It's a pretty straightforward one. So let's consider that population of beetles. All right. And within any population, there's variation. In this case, we've got variation in beetle color. All right. And then, of course, there's more individuals produced that can survive. So many of them die. In the case of these beetles, they're getting eaten by a bird. All right. Now have a look at the background, have a look at the beetles. Obviously, some of these variations are going to be better adapted to the environment than others. In this case, it's not good to be very light in colour, is it, against a dark background? So they're the ones that are going to be picked off first by a visual predator like a bird. So these light-coloured variations are going to be eliminated, whereas the better adapted ones, the dark ones, presumably, they'll be more prevalent in the next generation and pass on those traits. So the survivors are going to reproduce, they're going to pass on those traits. So with every generation, we're going to get a population that's better adapted to their environment. In this case, we're going to end up with more dark-coloured beetles and fewer light-coloured ones. Now, of course, if something happens to the environment, so it changes, in this case, if it's changed to a very light colour, then the tables are turned, aren't they? The dark-coloured beetles, they don't have the advantage anymore. All right. So is everyone okay then with that sort of quick run-through of evolution, natural selection? Yeah? That's Darwinism in a nutshell. A tiny little nutshell. All right. So now I want to look at concept 4. All right? The process of science and the scientific method. All right? So this is how scientists do science. Now, I'm going to take you through quite a rigid sort of scientific process, and then I'll tell you really how science is. So I want you to know this sort of rigid steps of the scientific method, but then I'll show you sort of, how science is really done. It's not as rigid as how I'm going to portray. All right, so let's think about science and the scientific method, how science is done. So science is taken from, I think it's a Greek or Latin word, to know. I think it's a Latin word for to know. That's what science means. And scientific inquiry is about trying to explain the world about us. It's about trying to explain natural phenomena in the world about us. Okay? So, we might want to try to understand that strawberry poison dart frog. Why is it toxic? Why is it red? What are all the mechanisms that cause it to be red, cause it to be toxic? And of course, we need to develop technology, like microscopes, to help us answer these questions. And sometimes the technology stops us making progress, but with every technological advent, usually we make big jumps. As soon as we figured out how to sequence DNA, we made enormous leaps in so many different aspects of biology. All right, so this is typically how scientific inquiry starts. It starts with observations. We make observations about organisms, about nature, about cells, about effect of drugs on your body, but we make observations. And they can be at many different levels, the observations that we make. And this is the nice bit about science. It's really easy to make observations, but sometimes you've got to train yourself to make the observations. So I'll tell you who are really good at making observations. Poker players. Really good poker players. They can look at just the tiniest little body language or twitch up, and they know what's going on. They're really good at making subtle, fine observations. So do you see what I mean about training yourself to make observations? Poker players are really good. So are scientists. They sort of train themselves to observe things that you don't if you don't have that sort of training or at least that interest. So once you've made observations, they generally lead you to ideas. You come up with some ideas. All right? And those ideas, sometimes you'll come up with an explanation to explain the idea or to further develop the idea. And explanations for questions you might pose based on observations, we call those explanations hypotheses. So once we've come up with an explanation for a question that's based on an observation, then we go ahead and test those hypotheses. All right, so that's the sort of process I'm going to take you through. All right, so let's look at observations first. So anybody know who this is a picture of? Yeah, it's Jane Goodall. So she did a lot of her work on chimpanzees in Tanzania in a place called Gombe. All right, and she spent years upon years out in the jungle making observations on the chimpanzees. And she kept very careful, very detailed journals of all of her observations. That included words and drawings and photographs, but she was quite meticulous, and she wanted to understand chimpanzee behavior. Do you think her presence influenced the chimpanzees?
Oh, yeah.
Probably. But we don't really understand, I don't know, what her influence was. Yeah, initially it might have been quite great. They were probably really frightened of her, probably very standoffish, which they were. But then as they got used to her and recognized her face, then maybe she blended into the background and she didn't influence them. But you do got to think about that. Yep, how your presence influences your observation. Do you think you being next to a plant is going to influence observations you might make about that plant? Probably not. All right. Although of course there's some folks who think if you sing nicely to a plant, it will grow better, and if you sing meanly to it, it won't grow as nicely. So maybe there's some truth in that. I don't know. But making detailed observations, that's really important. So I want you to think about observations, and when you make observations, you're generally collecting some kind of data. Now, I want to split data into two mutually exclusive categories. You've got something called qualitative data. And qualitative data is very descriptive. So, for example, to use Jane Goodall, the way that one chimp groomed another, or who groomed who. Okay? Do you see how it's very descriptive? And there's actually some pictures out of her notebooks of some of the written observations she made and some of the pictures. All right. So let's contrast this qualitative data, which again is generally very descriptive, with quantitative data. And quantitative data is usually numerical. So, for example, you might want to record the length of time of a grooming session. So let's just say that one of your observations was, it seems like the dominant chimpanzee is the one that always gets groomed by subordinate chimpanzees. That would be maybe some qualitative data. And then you might start your stopwatch and say, all right, each subordinate chimpanzee grooms for 10 minutes and then it's replaced. I don't know what her observations were, but can you see the difference between qualitative and quantitative data? You're okay with that? Yeah? Okay. So quantitative data being numerical, that's the sort of thing that we can plot on graphs and so on. We can also graph qualitative data. There's different ways to analyze and look at data, but I just want you to be able to make that distinction, qualitative from quantitative. All right, so the scientific process or scientific inquiry nearly always starts off with an observation. All right, so I want to take you through sort of the formal steps of the scientific method. Observations. That's what you usually start off with. All right, so scientists, biologists, they make observations about the real world. So those observations then usually stimulate a question. You want to ask a question about the observation you just made. So let's just say Jane Goodall was out with the chimps and she made an observation that the babies never get groomed. That's what she observed. So she might ask the question, why don't the babies ever get groomed? Yeah. So once you've asked that question then, what do you automatically do?
Try to find out. Try to find the answer.
You do try and find the answer, but before you go out and do any experiments,
You just make a assumption, you have created a hypothesis.
you do what?
You take a stand on which side of the issue, what your opinion is.
Okay.
Hypothesis.
You do, you generate hypotheses. So what you try and do, I just want you to be really clear what a hypothesis is. A hypothesis is a testable answer to your question or an explanation to your question. But it's one that you've put forth. Doesn't have to be true, doesn't have to be accurate, doesn't have to be correct. In fact, most of the time when we're doing science, we don't know the answers to the questions. It's why we're investigating it. So you do come up with these hypotheses which are testable, potential, plausible answers to the question. All right? So you put forth your best idea about an answer to that question. So that's the hypothesis generating stage. Again, in the formal process of science, the next step would be to generate some predictions. Oh, look at that. Is that a spelling mistake? Does that say prediction?
It says it's predication.
Predication. That's my bad. That's a mistake. Prediction is what you develop next. So you come up with a hypothesis, a potential explanation for your question. The prediction part is what you say is, well, if my hypothesis is correct, then this is what I would expect to happen under a certain set of circumstances. So I'll give you an idea for that. Let's just say that I made the observation that Native Americans used a particular plant, creosote, to cure little infections of the skin. They would take the leaves, they would scrunch them up and put them on the skin infection. Let's say I made that observation. What kind of question might that stimulate or beg? Sorry?
How much of it do they use?
How much of it do they use?
Why do they, where do they come up with the idea to use that?
Okay, why do they use it?
What's in the plant?
What's in the plant? Now, presumably they're using it because it's a tried and tested method to cure these skin infections, right? Yeah. So then you might want to say, well, what's in it which is causing it to clear up these skin infections, right? So then your hypothesis might be, so you've asked the question, what's in it? Your hypothesis might be, the leaves of that plant contain some substance that kills bacteria. That might be your hypothesis, right? So you might develop the prediction that if the leaves of that plant contain chemicals that kill bacteria, and I do an extraction of the chemicals from the leaves, then it should kill bacteria in a petri dish. Okay? Now, part of that is a prediction, because I said, well, if the hypothesis is correct and I do this, then it should kill bacteria. That's my prediction. Do you see how that works, how it all sort of fits together? All right. So far, what you've done is you've strolled around in nature, you've made observations, you've asked questions, you've thought about some hypotheses, you've posed predictions, but you've not done anything yet, have you? What's the next step? Test your hypotheses. Yep, experimentation. So then, in the example I just gave you, you would go out, you would collect leaves of this plant, you would make some chemical extracts of the leaves, and you might then get petri dishes, and you might grow bacteria on the petri dishes, and you might add the chemical extracts from the leaves. That would be your experiment. So, what's the next part after you've done your experiments?
Reflect the results.
You collect the results, right? Collect the results. And you would analyze your data. Now, in this instance, your results might be, did the bacteria die or not? Now, it's not often that black and white, were they dead or not. You might want to look at, well, did it kill lots of bacteria or none? It might be a matter of degrees, right? But anyway, you would collect your results. And then what would you do with your results?
Well, they find a conclusion.
You would draw a conclusion. You would say, what do these results actually mean? So let's just say your results were, none of these bacteria were affected by the extract of the leaves. What conclusion would you draw? You would say, my hypothesis probably wasn't correct. Wasn't a good explanation. So, is everyone okay with that? They're the conclusions you might draw. So you would look at your results, see if they agreed with your prediction. In this case, your results didn't agree with your prediction. If your prediction isn't supported, then your hypothesis is probably not correct. Right? At least within the confines of the experiment you did. But let's just say that, killed all the bacteria. Killed them. What conclusion would you draw then?
Is there a bacteria in the leaves?
Your data supports your prediction, so therefore your hypothesis is probably correct, or at least it supports the hypothesis. Okay? And it just so turns out that we do have a plant growing in the desert, there's loads of it on campus, that does have chemicals in the leaves that do stop the growth of bacteria on a petri dish. It's called creosote bush. And the active chemical in the leaves, you ready for this one? It's a bit of a tongue twister. It's called nordihydroguaretic acid. All right, and it's quite a potent antibacterial. But it doesn't kill all bacteria. Okay?
You said creosote?
Creosote bush,
That's
yep.
the one that has that smell of
That's it. Smells nice after it rains, what the desert smells like after it rains. Okay? So it doesn't kill all bacteria though. Take a bacterium like E. coli, doesn't have too much effect on it. Take a bacterium like Staphylococcus, and it does, kills them. All right, very effectively. So you might come up with results that lead you to draw incorrect conclusions, kinda. So if you just tested the extract against E. coli, you might come up with the results, oh, there's nothing in it that kills bacteria. But it might be the kinds of bacteria. Yeah, question?
With that bush and the stuff in it, would that like harm you or anything if you were to like chew on it?
So, here's the deal with it. It's been under quite intense research for about the past 2 decades because all these chemicals in the leaves they found that there's some anti-HIV properties, some anti-cancer properties, antibacterial, antifungal, not just HIV but other viruses. So you might think, oh my goodness, I should chow down on that plant and I'm going to be fit and healthy for the rest of my life, right? So there was one study a long time ago which showed that there was some liver toxicity if you took the plant. And again, that study's been questioned, but as a result, it's generally not considered, people are like on the fence really about whether it's something that's good to take or not, because of that one study that showed the liver toxicity. But I tell you this for nothing, it tastes terrible. All right? So if you do chew on it, you're not going to have a smiley face. But people are drying the leaves and putting it into capsules and taking it that way. What we usually do, though, is we find these bioactive chemicals in plants and we extract them and we purify them and then we test them to make sure that the side effects, I guess, are small relative to the benefits.
But wouldn't that plant, if you took enough of it, it's going to start killing good bacteria?
Yeah, it might. It may well do that. When you're on antibiotics for a long time, you start to kill your gut microflora, and then other bacteria come in and the balance gets thrown off and it creates problems of itself, sure. All right, but is everybody okay with these steps of the scientific method? Now this is a very sort of formal, I guess, breakdown of the steps of the scientific method. Are we out of time yet? We're good, aren't we? All right. So I'll go through this example. The example's in your book. Again, it's not a biological example, but I think it illustrates those steps really nicely. So let's say you make the observation, flashlight doesn't work. You might ask the question, why doesn't the flashlight work? And you might come up with a couple of ideas, a couple of explanations. For example, you might think, the batteries are dead. That's a hypothesis. Or you might say, the bulb is burnt out. That's a hypothesis. I want you now to come up with some more hypotheses. Batteries are dead, bulbs burnt out. Come up with some more. Go on, be a little bit more specific about the wiring. Maybe something's been disconnected. Connection's has come undone in the flashlight. Give me another one.
You didn't turn it on.
Sorry?
You didn't turn it on.
So that's interesting. The assumption is that you've operated it correctly. And it's a real flashlight, not a figment of your imagination. You've not been hypnotized, right? So we do have certain assumptions. Yeah, we're assuming that you really did turn it on.
maybe it was dropped or something in water. I don't know. I put the flashlight on.
That's okay if you drop it and break it, but now tell me why it doesn't work.
Oh, okay.
Be specific. Come up with some other hypotheses.
The lens would crack.
But the bulb would still come on.
Could be the coil and the bulb isn't connected properly.
So wouldn't that be the bulb is burnt out?
Battery
Yeah.
could be in backwards.
Good. Batteries are inserted incorrectly. That's different, isn't it? Give me another one. It's 4 good ones we got.
The metal could be corroded. So
The metal could be corroded. Now it's kind of similar to the wiring thing, right? But it's a bit different to a connection coming undone. There could be corrosion in a terminal when a connection's not made. So we've got 5. Got some more.
Water damage.
But be specific. Say, tell me why it doesn't work. Water might have damaged it, but what damage has been caused? It's got to be testable. We can test the batteries being dead. We can test the bulb being burnt out. We can test the batteries being put in the wrong way. We can test there being a connection undone. But dropped in water, water damage. We can't really test that.
What about like the lid's not screwed on all the way?
All right, so there's a bad connection. So there's lots of different sort of versions of the bad connection. Lid isn't screwed on. That's okay. That's testable. I want another one.
Button is broken.
The on switch is broken. Switch is broken. All right. So that's again another sort of category of the bad connection. Give me another one.
Made in China?
That might be the reason why it's not a good flashlight, but it's not a testable hypothesis. There are more.
Could be something wrong with the housing.
Again, make it testable. Be like specific as you can.
Maybe you don't have the right kind of bulb.
Maybe the incorrect bulb. Good.
Well,
That's
batteries,
number 8.
You could go batteries that way.
Incorrect batteries. Good. Number 9. Come on, let's get an even 10. One more. One more.
Could be so many things wrong with this battery.
Maybe you have like the wrong like battery cap.
Okay, so a component of the flashlight's been incorrectly assembled. All right, have we exhausted it? Have we exhausted every possible reason?
Yeah, I don't know.
Yeah.
Sure.
Oh, no.
Well, are you going to screw the light bulb in? It has a little dent. You can't screw the light bulb in.
All right, so the contact isn't made all the way from the light bulb. That's testable. Slightly different to the bad wiring or bad contacts. All right, what limits your ability to come up with hypotheses? Because I think you're limited out now, aren't you?
Maybe your understanding of what you're trying to
Yes.
test.
Your understanding of the topic of investigation limits your ability to come up with hypotheses. It does. Now, you all have a basic understanding of flashlights, don't you? Yeah. So if I got in, I don't know, a fancy piece of equipment from the lab, and I said, I don't know, why doesn't this PCR machine work? You're like, well, I don't know. Just plug it in, turn it on. Yeah. You don't know anything about it. You don't understand it, do you? So it's kind of like the human body. Why doesn't this human body work? Ooh. There's so many things that could possibly be an issue, isn't there? And we have a good understanding of some things and an incomplete understanding of others. But what is true for sure is your ability to make good observations, ask good questions, and come up with good hypotheses is limited by your knowledge base. All right? Expand and increase your knowledge base, and you will be much better at making observations, coming up with questions and hypotheses. You've all seen the TV show House, right? Yeah. His knowledge base is enormous. He comes up with the craziest hypotheses, doesn't he?
And he's right most of the time.
Yeah, he's right most of the time. He's really good at what he does. He's got an incredible intellect and an incredible ability to put 2 and 2 together and make observations which your average doctor can't. Now, you know he's British, don't you? Did you know that? Isn't that a great American accent he does? It's awesome. It's much better than mine. All right.
What's your
Yeah, I kind of.
Oh, it's terrible. After a few beers, I can do it. I can cuss, but I'm recording my lectures, so I'm not going to cuss right now. I can cuss with an American accent. That's fine. All right. So here's what you do with your hypotheses then. All right? Is you want to test each of your hypotheses. So before we do our testing, though, we want to go through this prediction step. So let's take our hypothesis number 1. If the batteries are dead and we replace them with good batteries, it should fix the problem. The flashlight should work. All right? So in this case, our test is to replace the batteries, and we're going to get a result. Either the flashlight's going to turn on or it's not. So if the flashlight did work, we would say our results match our prediction, the flashlight came on, and so therefore our hypothesis that batteries were dead is supported. Now in this case, the flashlight didn't work. So our result contradicts our prediction, so our hypothesis is not supported. So then we would go ahead and test another hypothesis. Maybe we'd jump to the one where the bulb is burnt out. And again, if the bulb is burnt out and we replace it with a good bulb, then, here's our prediction, the flashlight should work. So we're going to replace the bulb. Here's our result. Flashlight works, so our hypothesis is supported. What would we do if the flashlight still didn't work? come up with a new hypothesis, right? But think about this. Let's just imagine you really did get a bum flashlight and the batteries were dead and the bulb was burnt out, right? You tested this hypothesis and you drew the conclusion, that wasn't the result. The batteries weren't burnt out. So sometimes you've got to be really careful, right? Sometimes there are multiple things going on and you've got to be aware of that when you're going through this very sort of like rigid process of the scientific method. All right, is everybody okay then with this sort of rigid steps of the scientific method? Do you think if I gave you a statement and I said, what is this, an observation, a question, a hypothesis, or a prediction, you'd be able to do that? Yeah? All right. So, can science answer everything about everything? Not really.
Not yet.
Well, not yet. And there are some questions, honestly, that are outside the realm of science. So what about if one of my hypotheses for the flashlight was, there's a little gremlin. He gets in there. Every time you turn it on, he just pushes the battery away from the contact. What about that one?
Kind of hard to prove that.
Don't rain on my parade. That's my hypothesis, right? That's the one that I like. Why not? Why isn't that a good hypothesis? How do you know? How do you know there aren't any little gremlins?
The hypothesis has to be testable.
It's got to be testable. Right, in the case of little gremlins, I guess if there's really a gremlin there, you should say, all right, I can record it, I can see it, I can measure it, detect it somehow. But if there's no way to do that, it doesn't mean to say it doesn't exist, but it's outside of what we're able to test. And if it's not testable, then it's outside of what we can answer in science. Right? So, have you ever gone down to the bottom of the garden and seen mushrooms there? It's difficult to do in Arizona, but if you live in a place with lawns, you'll sometimes see mushrooms down the bottom of the garden, right? How'd the mushrooms get there? No.
Well,
It's obvious. Fairies put them there. They sprinkled some fairy dust and the mushrooms grew up. That's my hypothesis. No good? Not testable, right? So it's why things like, does God exist? It's outside the realms of science. It's not a testable hypothesis. All right? So in some ways it's kind of a silly argument to say, well, can you scientifically prove the existence of God or disprove? All right? It's outside the realms of science to answer those things. Do your beliefs and your opinions, do they factor into scientific outcomes?
I think so.
You think so?
Sometimes you don't account for certain possibilities because you wouldn't consider it.
So the answer is they shouldn't, but they frequently do. All right? Okay. All right. So I want to go over a case study then of scientific inquiry. It's kind of a nice one. All right? The data is pretty nice. So it has to do with the colour patterns of the coats of animals in nature. All right? So here was a study that was done. There were two mice populations of the same species. It's a species of Peromyscus. It's a little mouse. And they had different coat patterns in different environments. All right? There was a white-coated mouse that lived on a beachy area that had very light, almost white-coloured sand. Makes sense, well adapted to its environment. And then inland, there was a dark brown-coated population of this mouse in like a wooded, forested area, which had dark soil. And that makes sense, doesn't it? Yeah? So, the beach mouse, white-coated, lives on white, beachy sand. The inland mouse, dark coat, lives on darker soil. So here's little pictures of the little Peromyscus. There's our light coat one on the light beachy sand. There's our dark coated one on the inland population. And there's a map showing where they are. It's in a place in Florida this study was conducted. All right, so at first sight, looking at these animals, you might not even think they're the same species, would you? Because they look so different. But those looks are very superficial. It's just coat colour. So we've got two types of mouse, and they match the colour of their habitats, which kind of makes sense. Now, the natural predators of these mice are all visual predators, right? There's a lot of birds of prey that are going to hunt these mice, and they're very visual predators. You're going to have a lot of small carnivores or omnivorous animals, foxes and so on, that are also hunt them, and they're going to use sight as their primary cue. So it was hypothesized that the color patterns had evolved as adaptations to protect the mice from predators. As you might think, if you're a light-coated mouse, you'd be well camouflaged on the light-colored beach environment. If you're dark-coated, you'd be well camouflaged on the dark environment, and you should have greater survivorship, right, if you're well camouflaged. So a group of students tested that hypothesis, all right, and here's the experiments that they come up with to test it. So, first they made the prediction, though, that mice that did not match their habitat would be preyed upon more than mice that did match their habitat. So how would you test this idea then? What would be one way you could test this prediction?
Switch mice up in different habitats.
So go get the mice, dark-coloured mice from the inland habitat, let them go on the beach, and then go get the light-coloured mice off the beach and let them go in the dark-coloured habitat. What happens when you let a mouse go? Boom! It's gone. You're never going to see that guy again, are you? Now, there are ways to do it. You can put out traps at night and bait them, and you can recapture released animals. I guess if you captured 100 of the light-coloured mice, released them inland, and then put traps out, and you could see, well, how many of those light-coloured ones would you recapture compared to dark-coloured ones that you had out there as controls, and vice versa. So that's not quite what they did. So they built models of mice, all right, and they painted them to match one of the surroundings. So they built models of mice, and they painted some white. They built models of other mice, and they painted them brown, okay? And they put control white mice on the beach, and their dark-coloured mice also on the beach.
So they were both models fake?
Yes, both fake, all right? And then they got models of their white-coated and their brown-coated ones, and they put them on the inland habitat. Okay? And then they recorded signs of predation. So imagine if you're a predator, right? You're going to see what looks like a mouse, you're going to dive in, you're going to get it. If you're a hawk, you're going to grab it, and then suddenly you're going to realize what? You've been duped, right? It's not real. But there's going to be some sign that you were attacked, and that's what they were looking for. All right. So this was the hypothesis they were testing, right? That the colour patterns have evolved as adaptations to protect the mice from predators. So if that's a good explanation, what would you expect to happen to the mice on the beach? You've got white-coloured ones and dark-coloured ones.
The dark ones would get picked up by either hawks or birds of prey. And then the ones inland, the white ones would be picked up.
Okay? That's what you'd expect to happen, wouldn't you? So here's what I want you to do. I want you to draw some graph axes. That look like that. So this graph is for our beach environment. This one's for our inland environment. Here we've got our artificial mice that are painted white or are light in colour. And this is for our artificial mice that are dark in colour. So they put out a whole bunch of white mice at the beach, and they put a whole bunch of dark mice at the beach. And then they went back, I don't know, a couple of days or a few weeks later, and they looked for signs of predation. So we'll have this y-axis on the graph be degree or sign of predation. Okay? Obviously up here is lots of predation, and down here would be none or little. Here's what I want you to do. I want you to draw in bars of the graph if that hypothesis is correct. I want you to predict what the results would be if that hypothesis is correct. Good? All right. So these graphs then are your predictions, right? So let's see what results they actually found. And obviously if the results agree with your predictions or match them, then the hypothesis is supported. The white mice on the light background, obviously you would expect very low levels of predation. In contrast, a brown mouse on a white background is going to stick out like a sore thumb. You'd expect it to get attacked an awful lot more, so you should have higher signs of predation. And then vice versa on the inland habitat.
Inverses of each other.
Sorry?
Inverses of each other.
Yeah. All right. And these are the results they actually found. Okay. All right. So what's this guy? Here's a barn owl. And I believe it's the world's most widely distributed bird species. I think it's found on every continent except Antarctica, I think. It's a cool animal. Has anybody ever seen a real live barn owl out in the real world? For being pretty common, you've got to be in the right place at the right time, and they're almost exclusively active at night. So lots of studies have been done about owl predation of small rodents, all right? And they've looked at it in a lot of different ways. So here we've got number of mice caught on this y-axis, okay? And here we've got light-coloured soil. And over here, we've got a dark-coloured soil, also the number of mice caught. Here we've got our light-coated mice and our dark-coated mice. And these data were collected at full moon and when there was no moon out. Okay? Now what I want you to do is to try and draw some conclusions from these graphs. All right? I want you to write down what conclusions you draw from the data in these graphs. Okay? So look at them, try and understand them and make sense of them, and then methodically and systematically go through and see what conclusions you can draw. probably write half or a whole page of conclusions just on this, on the data in these graphs. A lot of different things you can tease out. Right, before we look at these, how many of you are going into healthcare? Just stick your hand up. Right, guess what your jobs are going to be? Looking at data and drawing conclusions. It will be numbers, it will be charts, there will be a lot of data you're given, and you've got to draw conclusions, right? A really good healthcare worker, whether it's a nurse or a physician, is able to generate lots of awesome conclusions from the data, okay? And it's a skill that it's a really good skill to learn, all right? To tease out as much as you can from the data. All right. So does anybody want to share any of the conclusions they drew from these data? Any brave soul? What you got?
Based on the second graph, I wrote a question right next to this because I wasn't sure, but is it colorblind? Like, is it a colorblind owl?
No.
No? Okay, then I...
Just what I'm asking you to do is draw conclusions from these graphs.
Don't they have really good night vision?
Owls do,
Yeah, so.
but they've also got exceptional hearing. In fact, their ears are even slightly different, slightly offset.
Yeah, they can hear movement, or they can see and hear movement.
They can't see in absolute darkness, but they have very good night vision, yeah. So conclusions. What conclusions did you draw from these graphs? Anyone?
Well, I can tell that like they aren't colorblind. So you can tell that they see like
So here's the thing, even if they were colorblind, we're not testing if they can see color, right? Because we just got light and dark. They could totally see in black and white. Yep. I'm assuming they do have good color vision. I don't know much about the sight in owls. I'm assuming they do have good color vision. But this is simply about dark and light. What you got?
I said during the first graph,
Yep.
where there's a full moon and you have a darker mouse on a lighter soil, the contrast of the mouse versus the soil and the movement, if it's moving, it probably caught the owl's eye more naturally, versus a light on light where it was a little bit more difficult to see. So it's just slightly raised, versus if there's no moon, the light on light, they would be able to camouflage and blend in a little bit more, versus the dark,
Yep.
where it's going to be blatantly obvious, like
All right, good.
a black marker to a whiteboard.
Yeah, good. So you've taken that first graph and you've described the results that are up there. Yeah. On a full moon, on a light background, there's no advantage to being light or dark for the reasons that you've said. Yeah. The light one blends in, but it's moonlit. Presumably they're quite visible running around. They've got a shadow underneath them. And the dark colour one also sticks out. So on a moonlit night, not necessarily an advantage of having a coat colour that matches your surroundings. But no moon, oh my goodness. Bad news to not have a coat colour that's the same as your surroundings. Yeah? But look at what happens to the predation of the white mice. It also goes down on the no moon. Okay? All right, so what about the second graph then? Conclusions. What conclusions could you draw from that? It's
Kind of little of the opposite of what was happening with the light colored. The dark colored on the full moon, the light colored mice were being attacked more than the dark colored.
Yeah.
And then with the no moon, it's about a little even because of movement. So
Yeah.
it was listening to the movement, so it's going after the movement.
Okay, and even on no moon, there's going to be some light around, so presumably the light one's stuck out. Yeah? So this is an interesting set of graphs to interpret because you've got what's known as an interactive effect, right? The effect of your coat colour relative to surroundings differs depending on the surroundings. It's not the same on a moon and no moon night. Yeah? Was this a challenging graph to look at and interpret and draw conclusions from? It is. It's a challenging graph. If I gave you one, it's not nearly so difficult, right? If I gave you one or the other, you could probably describe and figure out what's going on. But the two of them together, it's unlikely, if I gave you this one, that you would have predicted what happened here, and vice versa. Yep? All right. So, do you remember one of the skills? I said one of the skills I wanted you to develop was scientific literacy. Yep? Part of the scientific method is your ability to look at these graphs and interpret the data and figure out what's going on, draw conclusions from data. That's a really important skill.